Good morning, everyone. My name is Pekka Simula. I'm the CEO for Herantis Pharma, a drug development company based in Helsinki, Finland. And I wish you all very welcome to this LE&RN online symposium with the subject title, Clinical Progress with Lymphactin, Could Gene Therapy Cure Lymphedema? So with that, to the actual content of this LE&RN online symposium, and Herantis Pharma's own disclaimer, as we are a public company, we always want to make sure everyone understands that anything we say here is not meant as an investment recommendation or recommendation to buy shares in our company. And disclosures. The human clinical studies presented here have been conducted under all the appropriate approvals and in compliance with all applicable rules and regulations internationally governing the clinical development. So first of all, quick introduction to our company, Herantis Pharma. We are a public drug development company focused in regenerative medicine, and we have two highly differentiated clinical assets in development. Lymphactin gene therapy for the treatment of secondary lymphedema is the topic of today's presentation. We are also developing a clinical stage therapy aiming to stop progression of Parkinson's disease. And the common factor in this both really is the strong science. Both of these are based on globally leading science in their fields. And our aim really is to develop regenerative medicine based on cutting-edge science for patients in need. We're listed, publicly listed as mentioned, in Nasdaq First North Helsinki under the ticker HRTIS. So lymphedema, obviously, as you well know, is a significant and growing unmet need. And I've taken here what I find a very important quote. The psychosocial impact of lymphedema has been described to be as distressing as the initial diagnosis of breast cancer. I think this tells quite a lot to people who are not aware of lymphedema and who don't really understand how an awful disease it is. Obviously, as the next slide tells, the awareness of lymphedema is rapidly increasing thanks very much to the hard work by LE&RN. We are all very grateful for that. And I believe the audience of this symposium knows very well what an awful disease lymphedema is and also how common it is. This is usually what surprises everyone. But I believe you all know that lymphedema is a chronic progressive swelling of tissues. It's caused by some problem in the lymphatic vasculature, and in specific, secondary lymphedema is often caused by cancer or some other disease, can be caused by trauma or surgery, and it is a disabling and disfiguring disease which causes a huge impact on the quality of life of the sufferers of lymphedema. Now, the topic of today's symposium is Lymphactin gene therapy. And to help you understand what this is about, I'll have to provide a bit of a scientific background to Lymphactin. I'll try to keep this relatively non-scientific so it can be easily understood by also people without a strong science background. Obviously, if there is interest in digging deeper into the science, we will be very happy to provide you with links to scientific publications and so forth. But let's start with the basics. What creates our lymphatic vessels? Because we all have them. And the formation of lymphatic vessels is called lymphangiogenesis. And this is promoted by a protein called VEGF-C, VEGF-C. That's a natural growth factor which we all have. So all humans have VEGF-C in their system. And that's the protein that triggers the growth of new lymphatic vessels. So in theory, when we have when we develop lymphedema, in theory it could be treated with this growth factor. You know, so there's a damage in the lymphatic system. So why don't we just increase the VEGF-C levels locally in the area where there is such a damage? The problem, however, is that just injecting this natural growth factor would not maintain high enough VEGF-C levels for a long enough time. So this would not result in a clinically relevant growth of new lymphatic vessels. It wouldn't be sufficient. And therefore, we have developed a Lymfactin gene therapy. And this really is a drug in the development that hopes to cure the underlying cause of secondary lymphedema. That is the damages of the lymphatic system that cause secondary lymphedema. And Lymfactin is a so-called gene transfer vector. It carries the human gene coding for this protein, this growth factor, VEGF-C. So, in a way, Lymfactin is a drug that instructs the patient to produce VEGF-C in the damaged area locally. So when we administer Lymfactin, we help the patient's own tissue to produce this growth factor that will then trigger the growth of new lymphatic vessels. And as compared to just administering the protein itself, the good thing here is that the expression of Lymfactin lasts for about 2 weeks. And that has been seen to be a long enough time to actually meaningfully trigger the growth of new lymphatic vessels. And so first there is lymphatic injection in the damaged area. And in this picture, the lymphatic vessels are shown in the green color. So there is a cut in the vessels. There is some damage caused here. So Lymfactin is injected in the damaged area, and then the expression of VEGF-C will trigger lymph angiogenesis. So as you can see, there is sprouting of new, very small lymphatic vessels. And that eventually will result in the maturization of these small lymphatic vessels into a functional lymphatic network that aims to cure the damage that has led into the development of lymphedema. The next slide shows some of the preclinical work that has been done over the past years with lymphedema. So obviously, before something like this is taken in clinical use, one has to prove that it is actually safe and also that it has the potential to actually benefit the patients. And this is a model of lymphedema in which Lymfactin, our gene therapy product, was injected in the ear skin of mice in different doses. So there is a control group in which the vector did not contain the VEGF-C gene. And then there was the active vector containing the VEGF-C gene, and that was injected in several different doses. And as you can see, when we go from 2 times 10 to the 6th to 2 times 10 to the 9th viral particles, that means actual particles of the vector carrying this VEGF-C transgene, there is a clear change in the sprouting of lymphatic vessels. So this staining, the green color in this slide, indicates the sprouting of lymphatic vessels. And then the same thing has also been done in a porcine model of lymphedema. And again, The panels A, B, C, and D show different approaches. So there are 2 control groups and then 2 different ways of administering Lymfactin, which is here by the name of AD-VEGF-C. So intranodal AD-VEGF-C, meaning that Lymfactin was injected directly in the lymph nodes, and perinodal AD-VEGF-C, which means that Lymfactin was administered in the tissue surrounding the lymph nodes. And as these results show, both of these significantly increase the number of lymphatic vessels connected to the lymph node compared to the AD-LAC-Z control vector. Also, as a result of this treatment, the size of the lymph node has increased significantly as compared to the control group. These are all published data, so if some of you are interested in further details, these are published in a peer-reviewed scientific journal. And that's really what we believe in. So drug development is always very challenging. There are always big risks in the development of any new drug. And we always believe in the strong science. All of this work leading to where we are today has been published in leading scientific journals. So the entire path from the discovery of VEGF-C, this growth factor specific to lymph angiogenesis, all the way to the development of the adenoviral gene therapy and its preclinical development for the treatment of lymphedema. Of course, we owe a lot to the inventor of Lymfactin, our advisory board member, Professor Kari Alitalo, is the director of Translational Cancer Biology Research Program at the University of Helsinki in Finland. That's a national center of excellence for years and years. And Professor Alitalo is really a leader in the world in these endothelial growth factors. He has more than 500 peer-reviewed scientific publications under his belt, and he is also a foreign associated member of the National Academy of Sciences of the USA, in addition to having received numerous international science prizes. So it's really a privilege to work with someone like Professor Alitalo in the development of a novel treatment for lymphedema. So that's the scientific part. What does that all then mean from the patient viewpoint? So if you are a lymphedema patient, what good will all this science do in practice? Well, Lymfactin has already advanced in formal clinical development, so we are already actually treating real lymphedema patients with Lymfactin. And if it works as well in humans as the data in the disease models suggests, then the patients will benefit from, first of all, forming of new functional lymphatic vessels, which is then expected to lead to normalization of the lymph flow, which eventually will reduce the swelling and hopefully to a cure of lymphedema. Obviously, when we say if, we have to remember that drug development is always risky. It's also slow and regulated. So the development of any new drug to the market can take from 10 to 15 years, starting from the very early research. And the nice thing for Lymfactin, obviously, is that we have already reached clinical phase 2. So we have taken Lymfactin beyond some of the biggest hurdles, and are already looking forward to actually seeing if it truly works in humans suffering from lymphedema. So while the development of new drugs is very slow and takes years and years, and while we have to remember that the safety of the patients is always the #1 priority, so years of preclinical studies are needed before you can treat the first patient. We have already completed all of that for Lymphactin. We have already completed the preclinical stage. So we have reached the clinical part. And again, usually in drug development, the first stage after preclinical studies is a phase 1 clinical study in which the clinical safety is initially assessed. And also this has been already completed for Lymphactin. And actually it has been done in real lymphedema patients, not just in healthy volunteers, as typically is the case in phase 1 studies. And finally, we have now reached the phase 2 clinical studies in which the efficacy of the drug is assessed. Perfect timing really by this webinar, because we have just today announced that we have launched a phase 2 study called ADEAL. And that is a phase 2 study in patients with breast cancer associated lymphedema. Briefly about the phase 1 study that we have completed already. Earlier this year, we announced positive interim data from that study. So this was a study done in breast cancer associated secondary lymphedema. The Lymphactin administration was done in combination with lymph node transfer. So these were patients who were independently undergoing lymph node transfer as a treatment of their lymphedema. And together with the surgery, the patients then received a single dose of Lymphactin gene therapy. This study recruited a total of 15 patients with breast cancer associated lymphedema. And in addition to a very good safety, essentially no side effects have been observed. We also observed some very promising quality of life changes in these patients. However, it's very important to remember that this phase 1 study did not have a control group. So even if we see any signals of efficacy, we have to take that with a grain of salt. It's uncontrolled data, and it's also common in early clinical studies that patients feel much better after the clinical study because they have been taken care of so well. So in clinical development, the efficacy has to be always showed in a placebo-controlled study. And that's exactly what will happen in this phase 2 study. ADEAL just launched, and ADEAL stands for adenoviral gene therapy for the treatment of LE. It's a randomized placebo-controlled and double-blinded phase 2 clinical study in patients with breast cancer associated lymphedema. There will be a total of 40 patients recruited in this study, randomized 20 patients to receive Lymphactin gene therapy and 20 to receive placebo. And this is a very common setup in the first clinical study aiming to establish the efficacy of a novel drug, because you really need to compare against placebo. And double-blinded in this case means that neither the patients nor their treating physicians know which patient is receiving placebo and which patient is receiving Lymphactin. So there is no bias whatsoever from any possible direction. These are also patients who will receive a lymph node transplantation. So half of the patients will have that combined with Lymphactin. Half of patients will receive placebo at the same time. And obviously the idea in this setup is that Lymphactin is expected to improve the results of the lymph node transplantation by really vascularizing the transplant lymph nodes in the axilla. In this study, efficacy will be assessed by several endpoints. Quality of life is obviously very important. We need to make sure and want to make sure that the patients receiving Lymphactin are actually doing much better, feeling much better. Also, volumetric measurements of the arms, lymphoscintigraphy, which is an imaging approach to visualize the functionality of the lymphatic vessels, and also magnetic imaging, and then a lymph scanner tool, which we hope to be a very good way of, in a non-invasive way, assessing the how bad the lymphedema is. We expect this clinical study to be fully recruited by the end of 2019, and then we'll have to be patient and wait for the results for an additional 12 months, because we want to make sure that all the patients will actually have had the benefits that Lymfactin can add. So first of all, the maturization of new functional lymphatic vessels, and then gradually the decreasing of the swelling as the lymphatic system is back to its more healthy status, and then eventually also reduction of the swelling. So we expect the unblinding and efficacy readout of this study to be available during the year 2020. So we've also discussed this approach with key opinion leaders globally, both in Europe and the US, and some reflections on how they see Lymfactin as a possible novel treatment for secondary lymphedema. We are very happy that the KOLs are as enthusiastic as we are about the potential to achieve durable efficacy. So a huge difference to patients having to wear compression garments every day or receiving lymphatic massage very frequently, into something that with just a single dose would achieve durable efficacy, really in the best case cure lymphedema. The KOLs also see that the delivery of VEGF-C with a gene transfer vector is a very attractive approach. They also agree with us that breast cancer-associated lymphedema is a very relevant starting primary indication for Lymfactin, and finally, that the quality of life measures are very important in clinical development. So if Lymfactin really works, it should also be able to significantly improve the quality of life of patients. So that's just a combination therapy in breast cancer-associated lymphedema as an adjunct to lymph node transfer. What about Lymfactin as a standalone therapy, and what about other lymphedemas? We believe that there is significant potential. So if Lymfactin works as an adjunct in breast cancer-associated lymphedema, then we have reason to believe that it would also work equally well in other secondary lymphedemas. And we are actually considering already another phase 2 study in which Lymfactin would be a standalone therapy, so not combined with lymph node transfer, but just a standalone therapy in any secondary lymphedema. And possibly this could be also combined with a removal of scar tissue, because we already have scientific data, even published scientific data, showing that Lymfactin triggers the growth of new lymphatic vessels also across incision wounds. However, scar tissue is more challenging for new lymphatic vessels to penetrate. So if there is tremendous scar tissue in the area of damage, then that might be a reducing factor considering the efficacy, the potential efficacy of Lymfactin. So, to summarize what has been discussed so far, Lymfactin aims at curing secondary lymphedema. That's obviously a very ambitious goal, but we believe it's with an approach based on very exciting cutting-edge science and the natural mechanism repairing lymphatic system, we believe that that's the right goal for us. It also is very well aligned with the goal of LE&RN, a world without lymphedema. We believe that this can be a great significant step toward that, reaching that goal. We are currently in a phase 2 clinical study in breast cancer-associated lymphedema. The initial safety of Lymfactin has already been established in a phase 1 clinical study. And very importantly, this work is based on globally leading science. And we are very, very grateful for the good collaboration with Professor Alitalo. Anyone interested in further information is welcome to visit our website. And we also have an introductory video on Lymfactin, which again explains briefly the science and the practical approach in Lymfactin. So thank you very much for your attention, and let's see if we have some questions by the audience. The first question is, can Lymfactin be used in primary lymphedema as well as in secondary lymphedema? That's an excellent question, and we do believe that there are certain primary lymphedema indications where Lymfactin could be used probably as a combination with certain other approaches. For instance, we have patented the combination of using CCBE1 and our Lymfactin, which could be a possible treatment for instance, Milroy disease. But frankly, that's a step further down in the future. So first, we need to make sure that it is efficacious in secondary lymphedema, and we believe that breast cancer-associated lymphedema is a very nice, homogeneous patient population in which this efficacy can be established. We are confident that if efficacy is shown in this patient group, there will be many, many applications for Lymfactin going forward. Thanks, that's an excellent question. Another question about gene therapy. So we say that Lymfactin is a gene therapy. What does that actually mean, and could that be dangerous? Again, a very good question. Gene therapy is a relatively new concept. There has been clinical studies with gene therapies for a couple of decades, but we are just seeing the maturization of that field, and the first gene therapy products are now in the market. So the FDA and the EMA in the USA and Europe have quite recently granted the first market approvals for the first gene therapy products. And let's keep in mind that Lymfactin, when it's compared to many gene therapies, Lymfactin is a very simple gene therapy in the sense that it doesn't intend to change the genes of the patient. So it's not repairing any damaged genes in the patient as some of the gene therapies do. Instead, it simply expresses the human gene locally and for a relatively short time, just for about 2 weeks, only to produce a protein locally in the damaged area. So for a gene therapy, it's a fairly straightforward and simple approach, and can be theoretically considered very safe. Obviously, we have to remember though that it's only currently in phase 2 clinical studies, and drug development is always risky. So there can be some risks that will only be understood later in development. So far, however, we have no reason to any safety concerns. Next question: when do we expect Lymfactin to be in the market? That's obviously highly dependent on the results from this clinical study, the phase 2 clinical study. If the efficacy is strong, we'll be able to move forward much faster. If the results suggest that we, for instance, need to optimize the dosing, that would mean a bit slower progress. But we could speculate that everything going well, it could be in the market in about 5 years. So unfortunately not too fast for the patients currently suffering from lymphedema, but hopefully offering great hope in some years. Then there's a question about when are you planning to launch clinical studies in the US, and also a related question, when is phase 3? Phase 3, we'd hope to start immediately after the phase 2 readout. So as said, that's about in the year 2020, if everything goes well. And then phase 3 would also would obviously be an international clinical study. So that would most likely be done in the US and in Europe, and maybe also in some other countries. And we'd hope to start a phase 2 clinical study in the US already ahead of that. That could, for instance, be the study as speculated in other secondary lymphodemas, and in an approach where, for instance, just the scar tissue is removed and then that's combined with Lymphactin administration. And then there's a question of whether phase 3 could be open to all lymphedema patients. That depends very much. I mean, as you know, drug development is very highly regulated, and the regulatory authorities will want to see a convincing, compelling study setup. So that's always a discussion with the FDA on what makes sense. And also it depends quite a lot on how you can define a patient population that's homogeneous enough that that a meaningful number of patients is sufficient to actually compare the active arm against the placebo arm. So very difficult question to answer at this stage. I think it's still likely that the first phase 3 study will be in breast cancer associated lymphedema only, for the reasons explained. But obviously, if the phase 2 results are very strong, there would be interest in maybe expanding faster in other secondary lymphodemas. And then a question about why does it need lymph transfers? That's a good question. We don't actually believe that it needs lymph transfers, but that makes a setup in which it's the patient population is very homogeneous. And again, this comes from the usual challenges in drug development. If you have a randomized study with 20 patients on the active and 20 patients on placebo, and if you just take any secondary lymphodemas, then even though the patients are randomized, you might just end up by chance in a situation where all the patients, or most of the patients in the placebo group, are patients with breast cancer associated lymphedema, whereas most of the patients in the other group would be patients with some other trauma associated secondary lymphedema. That could happen just by chance. If the total number of patients is smaller, then of course the likelihood of something like this happening is greater. And then that could significantly impact your results, because for some reason it might be that this drug works better in one subgroup than in another. So that's the simple reason that we want to keep the patient group as homogeneous as possible. So if these are patients who are in either case undergoing lymph node transfer surgery, it means that they have a similar stage of lymphedema. It's because of the same reason, and the patient population is very homogeneous. So we are really comparing apples to apples, no matter what happens in the randomization of the patients in the two arms. Some further questions. Does it really matter or make a difference if you have lymphedema but never had cancer, and there is no way to understand what caused lymphedema? That's a very good point. we believe that lymphedema could help in a lot of at least secondary lymphedemas, but obviously remembering that Lymfactin is local administration, so it would need to be administered locally in the area where there is some dysfunction of the lymphatic vasculature. Then that place would need to be identified. So you know, if there is dysfunction of the lymphatic vasculature in your armpit or in your groin, and the location can be easily identified for sure, then I don't see why Lymfactin couldn't be also administered in that case locally in that area, and it could be possible that it helps. But of course, this is again all speculation because we don't even have efficacy data from the first phase 2 study. So we hope so, but it's still uncertain. And there's a question on efficacy again in primary lymphedema. And could use in primary lymphedema be considered off-label once Lymfactin is marketed in secondary lymphedema? We hope so. Again, I think that would be a very interesting clinical study at some point going forward. And of course, as soon as the efficacy is established in secondary lymphedema, and as soon as we can really be comfortable with the safety of Lymfactin, it will be much easier for instance, any Lymfactin specialist to consider off-label use, or even preferably consider an investigator-initiated clinical study where they could really investigate the efficacy of Lymfactin in this kind of a patient population. But again, we look forward to the future and hope everything goes well, so we will also reach that point in development. There's a question on the origin of the drug's name, Lymfactin. That's a very good question. We believe it simply sounded, you know, it sounds easy enough, lymphedema, Lymfactin, and no one else had picked that name for anything, so it was free to be registered. So no great story behind that, I'm afraid. And so I think those were all the questions we had. Thank you very much. Great to have so many questions. It's always kind of strange to be speaking to a computer when you really don't know if someone's following you. So thank you very much for the good questions. Thank you very much for following us. Thanks also for the highly supporting comments we received through the chat. And feel free to visit our website for more information. In the meanwhile, we also thank LE&RN very much for arranging this, for all their hard work to increase the awareness of lymphedema, because that's also what really enables us to continue our work, get our clinical studies funded, get the resources we need to move forward in this challenging field. So thank you very much, and wish you all a great day.